In this video, we're going to continue on with looking at structures in the cells that are out there. And we're going to focus on the transport or movement of molecules across this plasma membrane. And so here you can see some different examples in this cartoon. And we can see some molecules are able to pass through this membrane if they're smaller and less charged out there. You can see a little bit of water can move through this membrane. It is polar, so it can move through there, but it's not happy when it runs into that fatty acid layer. And so we've got these pores or channel proteins built into the membrane to allow the movement of molecules from the outside to the inside. And also remember, we can also have movement from the inside to the outside or exterior of the cell. In this figure I got from another textbook out there, but I liked how it summarized things. You can see that semi-permeable membranes that are out there with this phospholipid bilayer itself are actually going to control the movement or regulate the movement of molecules. And we can see a variety of different characteristics here. Various hydrophobic molecules that are out there are going to run into problems because they can't go across those charged outside layers right there. If you're small and uncharged or polar, sometimes you can go through just because of your size. But if you are large and don't have a charge, or if you are ionic, you are literally going to bounce off of that membrane there. So those molecules aren't going to be able to go across easily. And some of you are like, damn it, why do I have to learn this again? It's one of those sort of ideas that um, your instructors are going to look at. We can keep teaching students these ideas, and eventually they're going to go from rote memorization again to that familiarity where you not only understand but appreciate what's going on there. And so when we look at the movement of these molecules across the semi-permeable membrane, we find there's really two ways that molecules can move. You can have what's known as passive transport or diffusion of molecules, and you can also have active transport. If it's passive transport, you're moving from an area where the molecules are in high concentration to an area where they're in low concentration until they reach equilibrium. And passive transport is different from active transport because you don't have the use of energy. So you can think of passive transport as water flowing downhill. It happens naturally, and it's going to happen until these molecules reach equilibrium there. With active transport over here, the difference is you're actually doing the opposite. You're using energy to concentrate molecules and moving them from an area where they're low in concentration to where they're higher in concentration. So naturally, those molecules want to use passive transport to reach equilibrium where they're equal all over the place there. But in active transport, you're actually going to end up with the molecules being more concentrated. And you've run into this idea of the process of simple diffusion because molecules can diffuse through air. And so if you've been on an elevator with someone and they've ripped a nasty fart, those fart molecules started out concentrated next to their butt, and then they diffuse throughout the air there until they reached equilibrium so that everybody in the elevator could enjoy those fart molecules that have now spread throughout the elevator there. But with cells, you've got that phospholipid bilayer there, that plasma membrane, and so those molecules are moving from high concentration to areas of lower concentration naturally across that phospholipid bilayer until they reach a form of equilibrium there. When you look at this process, here's this idea of simple diffusion. You're moving down a concentration gradient. But if you're a molecule that cannot get across this membrane easily, but the cell still wants those inside, what can they do? They can use these channel proteins or porin proteins that'll open up a gate and allow those molecules to diffuse from high concentration to low concentration. And you can even be specific with your channel protein and only allow things that are the correct shape to trigger that gate to open. But notice in all of these cases of diffusion here, we don't see energy being used. It's passive transport. The molecules are moving from high to low concentration. So think of it as you've got a whole bunch of people packed into a room. You open up a door, and some of those people are going to be able to move out of that room until you reach equilibrium, until you have equal number of people in both of these rooms here. And if one of those molecules moves across here, another molecule will move across to make sure that you maintain that equilibrium on both sides of that membrane. So when we look at this, we've been looking at the movement of solute. Notice the solute. So what's dissolved in your solvent to give you the solution? And if we look at osmosis here, osmosis is different because rather than looking at the solvent or the solute molecules, now we're looking at the diffusion of the solvent molecules. And remember, when you're talking about cells and membranes, we're talking about water is the solvent. So here, if we look at these two examples, you can see you drop, say, a salt crystal into water, it's going to diffuse throughout. And so think like when you make powdered Hi-C with a punch. You drop it in there, and those molecules, we stir it up. That's active. We're moving things around. But if you let them sit in your fridge overnight, tomorrow you're going to have some juice that is uniform with the stuff that is spread out. They have equilibrated throughout there. But with osmosis, now instead of looking at the solutes moving across the membrane, we're looking at the movement of water across a membrane, but it's going to behave similarly. It's going to move from an area where the water is higher in concentration to an area where the water is lower in concentration until it reaches equilibrium. And so again, we can go back to our little animation and see there is diffusion moving back and forth. There is your facilitated diffusion, non-specific and specific, where you're controlling the type of molecules. Both of these are the diffusion of what's dissolved in the water. And with osmosis, we're looking at the movement of water across the membrane as opposed to what's dissolved in that membrane, or what's dissolved in that water. And when we look at osmosis and the movement of water, this is going to play a very large role when you're talking about cells, because again, your cells are composed with a massive amount of water on the inside, and they've got water on the outside with these dissolved solids or solutes right there. And so we can look at the strength of a solution there. And if you have a cell that is sitting in a solution that is the same strength, and we can see that in this little cartoon right here. You've got a 20% solute concentration on the outside and the inside of this cell. Well, if 20% of my solution is the solids that are dissolved, that means 80% of it must be the remaining volume is water right there. So here you've got equal amounts of water and solute on the inside and the outside. So there's really no net movement of water because it's already at equilibrium. And when you look at a solution like this, it's known as isotonic. So here, think iso is the same and tonic is strength. So this solution is the same strength on the outside and the inside. And when cells are floating around, say in a river or a lake, there's a chance that they could be in an isotonic solution already. But then when they move to a new location, they can have a different strength solution. And one of those that we can see right here is hypertonic solutions. And when you look at hyper, think hyperactive kids, they're too active. Well, here hypertonic is over strength solution. And so if you look at this thing, the solution on the outside of the cell right here has a difference in terms of the water that is on the inside. So your cell starts out with this solution of water right here, and then this solution of solutes in water. So if we look at this solution right here, the solution on the outside of the cell has less water and more solutes compared to the inside of the cell. So there you can see the inside of the cell is 80% water and the outside of the cell is 60% water. And so when we look at this, the solution on the outside is hypertonic. It's over strength because there's more solute and there's less water. And so if you look at that and think about water is going to move from where it's high to low in concentration. And so the net result of this is when a cell moves into a hypertonic solution, it's going to have the water moving out of the cell. So I always remember this because when I think hyper, I think you're sweating, and that's what the cell is doing right here, sweating, and it's losing that water. And if we look at this thing and think about the net effect here, well, in this picture, you can see there's a cell wall on the outside, and what's going to happen to the cell? It's actually going to crenate. As the cell loses water, your cell membrane is going to shrink further and further down right there. And you've seen this. If you've seen a plant that hasn't been watered, it's going to be limp because the cells inside the cell wall are crenating or shrinking right here. When you look at the opposite side of this, the hypotonic solution, hypo, think hippos are hanging out in water and there's less solute dissolved in there. And so if we look at this one, you can see hypotonic solution has more water, and inside of the cell, it has less water. So the water is going to move from high to low concentration. So here, the water moves into the cell, and your cell is going to swell up. So again, hippos are going to swell. You can also think of hypotonic, think hypodermic needle. It's going under the skin. Literally, hypo is under, and dermis is skin. Well, here, tonic is strength. So your solution is under strength in terms of the solute, which means you've got more water there. And if we look at our example with our cell right here, when these cells are placed into the hypotonic solution, the water is going to move in there. And think about your plant that was all limp because it wasn't being watered. When you add water to your soil around your plant, that's going to cause the water to move into your cells right there, and that's going to cause the cell membrane to start swelling. And in the case of a plant, it's going to run into the cell wall. If you're looking at a bacteria, it's going to run into the cell wall. So your cell is only going to fill up with so much water before you cannot have any more water going in there. So this cell wall is going to provide protection for certain kinds of cells that are placed into a hypotonic solution or that end up in a hypotonic solution. And that's going to cause osmotic lysis. So our cells that don't have a cell wall, if they're placed into a solution where there's way too much water, think drinking distilled water, you can actually have your cells exploding because so much water goes in them that the membrane is going to fall apart there. On the active transport side of this, what we've been looking at is diffusion of solutes and solvents moving from high to low concentration, and that happens spontaneously, that happens naturally. Active transport, you're moving your substances against a gradient, and remember, you're using energy for this process right here. So here we can see sodium and potassium ions that are floating around in the solution. They can't move across the membrane because they are charged. So instead, a membrane protein is going to grab onto them, and then that membrane protein is going to burn some energy in the form of ATP molecules in order to allow that transport to occur. And if you look closely at this, you can see our sodium looks like it's being transported to the outside of the cell, and potassium is being transported to the inside. So you can get transport going in both directions. It can be in a single direction or unidirectional kind of transport that's out there. So this is going on inside of cells constantly. They are adjusting to changes in their environment, whether it's passive or actively trying to maintain homeostasis. They're trying to keep the interior of the cell in a relatively constant environment as the exterior is constantly changing. And bacteria can do something that we can't actually do. Bacteria can do a form of active transport that's known as group translocation. And I've got a simple little cartoon over here. You've got a sugar, a glucose molecule, and it binds to this membrane protein right here, and the protein uses energy to push that glucose into the cell. And this is really the cell effectively eating. But that glucose might leak out slowly, and the cell doesn't want to lose that because it spent some energy in this process of doing the transport. So the solution that bacteria cells have evolved is they actually chemically modify that molecule as it's being transported across the membrane there. And so in this case, you add a phosphate and make glucose 6-phosphate, And remember, phosphates have charges, and that's effectively going to trap the glucose inside the cell because with a phosphate, the glucose will not be able to diffuse to the exterior of the cell. So bacteria can do this process of active transport that eukaryotic cells can't do. But in the same hand, eukaryotic cells can use transport mechanisms that involve using sacs or vesicles of membrane right there. And we've seen this process of endocytosis and exocytosis in other biology classes. Endocytosis can move particles from the outside to the inside of the cell by basically pinching off little sacs of membrane, and that's phagocytosis, or the cell eating. Pinocytosis, they are bringing in liquids and some of the other particles that are dissolved in that. And receptor-mediated endocytosis is something that our cells can do, where a specific substance binds to a receptor on the surface of the cell, and that's going to trigger this membrane to deform and basically pinch off your membrane to make this vesicle. So you are literally transporting molecules to the inside in response to a trigger on the outside of the cell. And this is something bacteria cells cannot do.